A very good morning to all. I am Karan Sharma with my co-host Dr. Chandni Sharma. On the behalf of organizing committee, we will welcome all the dignitaries in this inaugural ceremony of 5 days faculty development program on empowering educators for excellence in academia and research, held at Shiva Institute of Pharmacy in collaboration with Spoken Tutorial IIT Bombay.
Faculty Development Program offers valuable advantages like recent knowledge by the experts, provide excellent opportunities to disseminate research and making professional contacts. On this occasion, I would like to share one thought of late Dr. APJ Abdul Kalam sir. Learning gives you creativity, creativity leads to thinking, thinking leads to knowledge, and knowledge makes you great. I hope this positive thought infuse a new energy in all of us.
We are about to commence our event by lighting the lamp of knowledge and wisdom. Lamp lighting is supposed to be bring a lot of positivity in the entire atmosphere. This is a symbolic movement that signifies the dispelling of darkness and igniting of thoughts and enlightenment.
I would like to request our invited speakers, Professor Sanjay Chajit sir, Professor and Head Department of Natural Products, Naipar Mohali, Professor Pallavi Ahirao ma'am, APTI National Co-Convener, Women Form, CCP Mohali, Dr. Anup Kumar sir, Delhi Pharmaceutical Sciences and Research University, respected Engineer Purushottam Sharma sir, Managing Director, Shiva Group of Institutions, Engineer Ayush Sharma sir, Chief Executive Director, Shiva Group of Institutions, Principal Shiva Institute of Pharmacy and convener of this FPP, Dr. Saurabh Sharma sir, to kindly come forward for kindling the lamp of knowledge and wisdom.
Shubham Karoti Kalyanam Arogyam Dhan Sampadam Shatru Buddhi Vinashaya Deep Jyoti Namostute. आइए चलें अंधकार से प्रकाश की ओर, पृथ्वी से आकाश की ओर, पदार्थ से सूक्ष्म की ओर, प्राणी से परमात्मा की ओर, एक प्रयास अज्ञान से ज्ञान की ओर. A heartfelt thanks to our esteemed speaker for leading this auspicious meeting. Thank you so much, sir. Now, I would like to take opportunity to introduce Shiva Group of Institutions. Shiva Group of Institutions is well-recognized institute of Himachal in the field of education that imparts quality education and practical knowledge to each and every student to reach their highest academic and personal potential.
The Shiva Group of Institutions is run by Shiva Educational Society established in the year of 2006 at Bhumarni. Institutes which are established under the banner of Shiva Educational Society are Shiva Institute of Pharmacy, Shiva Ayurvedic College and Hospital, Shiva College of Veterinary Sciences, Shiva College of Education, Shiva International School and Shiva International Residential School.
Shiva Institute of Pharmacy established in the year of 2015, offering D Pharmacy, B Pharmacy and M Pharmacy courses in different disciplines like pharmaceutics, pharmacology and pharmacochemistry. Shiva Education Society constantly working towards excellence in shaping the personality of future citizens of our region as well as for our country.
Now, I would like to request Dr. Saurabh Sharma sir, Principal, Shiva Institute of Pharmacy, to please come on the stage for welcome address of the guests.
Thank you, Dr. Chandni and Mr. Karan for this wonderful introduction to our institute. Honorable Managing Director Sir, Engineer Purushottam Sharma ji, our Executive Director ji, Mr. Ayush Sharma, our esteemed guest, Professor Dr. Sanjay Jhaja, who is the Professor and former Head of Department of Natural Products, Naipur Mohali. Dr. Pallavi Rao, who is the Professor of Pharmaceutical Chemistry and APDI co-convener of Women Forum all over India. Esteemed faculty members and dear participants, it's my honor to welcome you all in this faculty development program. As we embark our journey to this faculty development program, it is very important to empower our educators who shape the minds of the future generation. As educators, you play a very, very vital, important role in creative, critical thinking and innovation, inclusion into our students. And that is very important. So equipping our faculty members towards the directions, towards the right cutting edge knowledge, cutting edge skills, recent advancements is very, very important. And keeping this in mind, this particular faculty development program has been designed where we are having, you see, multiple institutes, multiple universities, multiple research organizations with us who are participating in this particular program. And it is also in collaboration with the IIT Spoken Bombay Tutorial, which is a project towards the, you see, perfect communication skills to be integrated in our students. The sessions in this particular faculty development program will focus on as the natural products research, the zebrafish research, that is very important. We will be having a CSIR scientist with us, and he will be highlighting on the zebrafish research, that is the need of hour, and we are also in a plan to establish a zebrafish lab, research lab in our campus also. In addition to that, we have IPR experts who will highlight the perspective of the patenting and plagiarism also, what is the significance of plagiarism in the patenting and other IPR rights. Then we will be having various experts who will be demonstrating about the software simulations, ICT tools, and artificial intelligence in teaching, learning, and research. So keeping this in mind, this particular program has been designed. Now let us all come together to have this knowledge as a Charan Vanna to the Saraswati Vanna, which we have already done. And this is very, very important. The God worship is very, very important in every task. Now, when we are talking about the incorporation of the qualities, of the skills, of the pedagogies, of the recent advancements in our programs, then it is very, very important to work together. And if we work together, we can have a change in our education system. The next important point is, when we are working towards the next advancements, then keeping this in mind, the same has to be communicated to our students also, where it is all the skills that we are learning, if they are going to all the students, then it is going at the right direction, because the end point is that. And not that only, after that it is going to the placements, which is the skills are very, very important right now. So keeping this in mind, I wish you all to have this faculty development program with a fruitful, energetic, engaging, productive, and enriching experience. Thank you.
Thank you so much, sir, for sharing the roadmap of upcoming sessions. It is really motivational and we are feeling energetic and committed towards our professional activities. Thank you once again, sir.
Now I would like to welcome our esteemed guest by presenting a token of honor. For this, I would like to request Honorable Managing Director, Shiva Group of Institution, Engineer Purushottam.
Indian medicinal plants for this area of antibacterials, that is AMR, antimicrobial resistance and antiviral. That's why I have also mentioned this area, that discovery of efflux pump inhibitors against escape pathogens. You know that this is a panel of 6 bacteria. First two are gram-positive, the next 4 are gram-negative. So this is the panel that we use, the bacteria which causes different infections in hospital settings and otherwise also. In antimicrobial resistance, they have big role to play. They are mutated strains of these bacteria. So that's why it is very important to find the strategies to overcome the antimicrobial resistance from natural products. So that was one of the important area which will be told in the second lecture by Dr. Pallavi. So I'm not going to tell much in details here. So these are some work that we are doing in collaboration with CSR Intech in Chandigarh over the last 10 to 12 years. We had several preclinical candidates from natural products, that is from plants, as well as microbial strains that they have in Intech. So this is what the 6 bacteria I mentioned that you can see escape. First 2 gram-positive, the next 4 are gram-negative. And efflux pump here are the most lethal weapon bacteria possesses to develop resistance. This is one of the mechanisms. This is not the sole mechanism. There are 3-4 different mechanisms more which causes resistance in bacteria, but this is one of them. And here, while working in collaboration, the collaborators lab, Dr. Himraj and Nandarlal, they isolated 2 simple molecules from microbial strains which shows actually efflux pump inhibitory activity. So we modified them as per the, let us say, their chemistry and did the semi-synthetic derivatization on those and did structure activity relationship studies where we find very interesting molecules. So with this RP1 and RP2, I will tell only here about little about RP2. This was the compound which was RP2 isolated from a microbial strain. So we can very easily synthesize it, that we have done it to get it in gram scale and then derivatize it further. So we have made different shift bases and other derivatives which I am not sure. Only the most active derivatives that I am showing here. And these compounds, some were very active as EPI inhibitors, efflux pump bacterial inhibitors against MRSA strains, as well as they are also showing very good antimicrobial activity. So you will see that SMJ3 and 5 are very good APIs, the best one and the positive controls. And the next 2, this in the same table, SMJ2 and 4, they are strong antimicrobials. So I will not go into detail. This work published in Communication Biology and Nature journal, where we have showed that these are actually broad spectrum antibacterial agents against multi-drug resistant gram-positive bacteria, including MRSA. So all the biology done there, chemistry we have done, and we could see very interesting result that I am just mentioning here in the last slide here. See, both these compounds, they were tested against both bacteria in vitro as well as animal model, and these compounds, they actually showed the activity on the growth and metabolism of S. aureus ATCC 29213 strain, including the accumulation of NADH, mRNA expression of NDH2 gene, and breakdown of this iodonitro chloride into pharmaceutical product with which we can check the activity. And these actually were very potent preclinical candidates that we have that can be further developed or IND can be filed from these 2 compounds. So that was one of the research area. So now I will switch gear coming towards the nutraceutical and phytopharmaceutical that we are working very actively in my lab since last 5 years. So I will not go into this details definition. You can see, you know that there is a paradigm shift from cure to prevention. This is what we talk about Ayurveda also. Ayurveda relies on the basic principle of prevention and cure. So the 21st century medicinal herbs are gaining importance in the mainstream healthcare as greater number of people seek relatively safe medicines. And demand for herbal medicines, herbal health products, herbal pharmaceuticals, nutraceuticals, dietary supplements, and even herbal cosmetics or nutri-cosmetics, that is the upcoming area, is increasing globally due to the growing recognition of these products as mainly non-toxic, having lesser side effects, better compatibility with our physiological system, and even the transcripts of traditional medicine systems that includes Rigveda, Ayurveda, Charaka Samhita, Sushruta Samhita. All these are very important texts which we can explore, or even different Ayurvedic institutes are doing a lot of research in that, and we are having collaboration here with those institutes to explore the potential of Ayurveda, that transcript classical formulations we can study in the light of modern medicine. That is what need of an hour. And of course, India has been known to be the rich repository of medicinal plants. As we are sitting here in Himachal Pradesh, you know that it has a very great or one of the hotspots of medicinal plant biodiversity. We see in western as well as eastern Himalayas. That's why, and even in western Ghats in the south. So our country is a huge country with big potential for the biodiversity and biodiversity of medicinal plants as well. So Hippocrates has already mentioned, you all know that let food be the medicine and medicine by the food. So we need to see food also from medicinal perspective apart from the nutritional needs of our body. So Ayurveda and nutraceuticals, the uses of nutraceuticals as advocated by Ayurveda can be classified depending upon the age of the individual, season of consumption, time of consumption, physiological conditions, and according to the target organ. This is very important because you see the seasonal, whatever you eat, the nutrition, that is always best than eating the food which is off season. So that is actually already advocated in Ayurveda, very important thing. Because the balanced diet is very important for the health of an individual. Then we all know that these free reactive oxygen species, ROS, they create oxidative stress, and that is responsible for several lifestyle diseases we see these days, starting with cancer, diabetes, obesity, and so on. That you see, these are the major diseases under metabolic syndrome X that we call. So here also there is a lot of need to do research in these lifestyle diseases and development of products from this. So these are the three categories we talk about: functional foods, nutraceuticals, and pharmaceuticals. So you see the basic difference there. Example wise, probiotic is a functional food. For example, Yakult, and there are so many on the market these days. Probiotics as well as prebiotics are there. Then nutraceuticals, one can say aloe vera gel, and even the conventional, I need not to tell, we all are from pharmaceutical background, all API based drugs, formulations are under pharmaceuticals. So this is the chart that will tell you how to develop a nutraceutical product that you can see. Depending upon the market availability, you find out the therapeutic segment that we are talking about. Today I will give the example of obesity and obesity associated with diabetes, that is diabetes. So that is the, let us say, therapeutic segment, and accordingly you try to find what is your materials that you will investigate, like the food matrix selection, I mean to say that materials. And then there we start the work, try to prepare the enriched fraction, and those enriched fraction you can fortify with different proteins and make a nutraceutical product through several approaches that we do in drug discovery, like you see bioaccelerated fractionation, we do in natural product research, and then finding the mechanistic, let us say, aspect of that isolated bioactive constituent that we can do, and at the same time we should know what are the different secondary metabolites that are present in those particular active fractions by using the modern methods of LC-MS that we generally use. Apart from HPLC UV, we use HPLC-MS extensively, and then we identify compounds once they are isolated by mass spectrometry as well as by the NMR spectroscopy. NMR that includes one-dimensional as well as two-dimensional NMR, and then of course the preclinical safety assessment that one has to do, and proof of concept of efficacy with respect to the, let us say, dose selection and all that you have to do. And then if required, like I mean to say, if you want to go for a single fraction based product development, then you may also require to do clinical trial. But as per FSSAI regulation, you need not to do clinical trial. for the nutraceutical product development. So this is the regulation which is given FSSAI 2016 and again modified in 21. These are the guidelines that are available on the Health Ministry website under this agency FSSAI. I have given the link that one can explore, but here the nutraceuticals, what is the objective is that it should provide a physiological benefit and help maintain good health. That is very important. And then further, a food business operator, one can prepare and sell the nutraceuticals in the food format of granules, powder, tablet, capsule, liquid, jelly or gel, semi-solid and other formats. And as well as we can also manufacture them and dispense them in sachet, ampule, bottle and many other dosage forms. So especially the section 4 of this nutraceutical regulation, what I want to tell is that it lists the 200 medicinal plants where we can take them for nutraceutical product development, and this is very important aspect that you people should check in this FSSAI regulation under Ministry of Health. So some superfoods like when we started work in our department some 10 years back to find the different medicinal plants and their active ingredients against obesity. So there we selected almost 34 medicinal plants, from them 102 extracts were prepared that were initially screened for pancreatic lipase inhibitory activity. You know we have a drug in the market, Orlistat, which works by inhibiting pancreatic lipase. So that was our initial assay, enzyme-based assay. Then we developed the cell-based assay that I am going to show later on, that is PT3L1 adipocytes, where we can study anti-obesity activity. And in that, among 34 plants, these were the most active plants. You know licorice, Lycyrrhiza glabra, as well as the curry leaf, Muraya conigli. These are the two different plants that we got as a lead. So this, I need not to tell, you are aware of this, but you know that this was also very popular during COVID time. It was the ingredient of many immunomodulatory preparations, is Lycyrrhiza glabra, and we have isolated 19 compounds from the rhizomes, that different extracts we prepared of licorice, and in vitro studies were done, and one of the active ingredients is a very simple compound that you see on the slide, chalcone, that is isoleucivitigenin, which is present in considerable amount, as well as I haven't shown the structure of nicochalcone 8, that is another major bioactive compound in this plant that shows anti-obesity activity by inhibiting pancreatic lipase as well as showing anti-adipogenic activity in adipocytes. And then apart from that, literature shows lemongrass that we use in our herbal teas also, or in our routine tea we use actually lemongrass. That also is one of the superfoods. Then we have curcumin. I will not talk about much here. We did a lot of work on curcumin, and where you will find many researchers are there on curcumin in the form of formulations like nanocurcumin is already available in the market, which is water soluble. Otherwise, curcumin is not water soluble. So these are some examples. Then you know omega-3, omega-6 fatty acids, fish oils, and even these chia seeds. They are actually the rich source of this omega-3 fatty acids, and they show very strong antioxidant activity. And by virtue of that, they show anti-obesity and several other health benefits when they are given as unsaturated fatty acids, omega-3 and omega-6 in different foods. Now I am coming to the next category of formulation, that is phytopharmaceutical. Now most of you might have read this regulation. This is first time in the world India has actually brought out this new drug category called as phytopharmaceutical. Let me tell you, as we are speaking, in 2020, USA also has started botanical drug standard. But there they ask 2 markers. India, we are getting 4 markers. Now this is a difference as with respect to US FDA and our CDSCO and DCGI, the drug authorities. So this particular is actually new drug regulation is to promote research from single medicinal plant. This is not the polyherbal formulation. This is the formulation prepared or product prepared from a single medicinal plant that you will see here. It is mentioned that it is a purified standardized fraction of medicinal plant extract with at least, previous slide, with at least 4 bioactive compounds used for disease diagnosis, treatment, mitigation or prevention. So Ayush launched drugs must meet reference standards with phytopharmaceuticals requiring both non-clinical and clinical data development. And now you see here, for a drug to be classified as phytopharmaceutical, there is a lot of stress that we have to put, that is available information on the selected medicinal plants for product development, formulation and route of administration, dosages, therapeutic class and all other relevant details. And one has to generate the data on identification, authentication, source of plant, used for extraction and fractionation, process of extraction, subsequent fractionation and purification, formulation details of phytopharmaceutical drug and the stability data. Now very important thing here is, you see the challenge lies in preparing a purified fraction. This is not the hydroalcoholic extract you prepare from the plant. Hydroalcoholic extract can contain up to 200 compounds. If you see, that is what our experience, what I see, every extract when we prepare, first thing we put it into LCMS. Unless and until you do that, you will not know what it contains. This is very important. So by seeing that, when we talk about extract, that's what this regulation don't talk about extract. It talk about the purified fraction, enriched fraction. And this purified fraction preparation is never easy because you have to have the certain chromatographic process which you have to, let's say, innovate and try to put new information where you can file a patent. Here you have a possibility to file process patent as well as product patent. And one example I will show later on. But what I mean to say, that enriched fraction will be a purified extract, which will have, let's say, 20 to 30 compounds maximum, not more than that. Extract has almost up to 150 to 200 compounds. And that is where the challenge is, that we have to show this enriched fraction and the batch to batch variation. Reproducibility is another challenge that we are discussing at national level. Where are the issues? Because as I am speaking, not a single product yet approved by DCGI. Why? Reason is this batch to batch variation, variability, but at least now they are ready with approval of 3 products from CSIR. Because all these concepts started with CSIR and NIPERS when Dr. Shekhar Mande was DG CSIR at Delhi, and they started this phytopharmaceutical mission under Department of Biotechnology, then ICMR, CSIR and DBT. They actually had the MOU in 2016 to promote this particular new drug category, and now we have 3 products ready for approval. IND has been filed. So this is where now you see this is a great sign that if you want to work on a single medicinal plant and that will be regulated like your conventional pharmaceutical product. So here is the drug category. And this one particular formulation, the active ingredient we say API, here we call it as APPI, active phytopharmaceutical ingredient. So that is the terminology. We have done that, and we are already giving the lot of the suggestions to Indian Pharmacopoeia Commission how to brought the guidelines to actually reduce the variation. So how much of the variation should be allowed and all that. That discussions are also going on currently. We are actively involved with Pharmacopoeia Commission Ghaziabad on the, let us say, redefining and giving newer insights in this new drug regulation that was notified in 2015, so 10 years down the line. Now we are ready with the final guideline, and we will see that a lot of drugs potentials are here. Many drugs will come from this category in near future. That will be actually a leading example in the world how the drug can be developed from a single medicinal plant. So this is phytopharmaceutical, very important that we are talking about. So this is the details you can read from this, let us say, website link. So what is important? So this is in line with regulations in USA. I already mentioned botanical drug standard that is called in USA. China already had this, and other countries like artemisinin that is coming from China. They talk about the phytopharmaceutical drug only, and that is a big example. If China can do it, why we can't do it? Definitely, I think this new drug is due from either Ayush or from this group in India, that we will see a blockbuster drug coming from India in the next few years. And this is what the very exciting opportunity all we have to develop a drug from a medicinal plant. So this is the example. So it is the 60 years hard work by this lady Yu Tu in China, where she actually examined several medicinal plants, more than 1,000. Out of them, one was Artemisinin annua, which actually gave the drug molecule artemisinin and its semi-synthetic derivatives. We know artemether and artemether that are used clinically for the drug-resistant malaria. So this was the phytopharmaceutical from China, got Nobel Prize. So I think one is due from India very shortly. That is what we are hopeful. And this is the area I think many people from pharmaceutics, pharmacology can also venture into. So we are working on actually this particular last example. I will spend 10 minutes and show how the product can be developed from a very common medicinal plant which is already displayed in FSSAI regulation, and of course it is already mentioned in Ayurveda also. It is called as Mithani. Kadu leaf, you know, Azadirachta indica. This is Mithani, Muraya conigi. Curry leaf that we use as a spice very commonly. In our kitchen, you will see South Indian dishes. They are incomplete without curry leaf. So this is curry leaf, all the literature that we compiled before we started also the work. And then as per the definition of phytopharmaceutical, we need actually enriched fraction and 4 markers. So we have prepared alkaloid enriched fraction from the leaves of Muraya conigi, that is curry leaf that we plucked from the plant. So those were dried, and we did the large scale extraction at pilot scale, and the trials were done at Nipah itself. Or we are ready with the everything preclinical safety, efficacy, only clinical study. We are starting next month in Dhanvantari Ayurvedic College in Chandigarh. They are already the protocol is placed in Humanities Committee, and once we have that data, 120 patients, double-blind, let's go clinical trial. We are going on this. Once that data is ready, 2 companies are already in talks. They are interested to take this product. So we'll license it as per our government guidelines, and this will be, I think, a major product from Nipah from Department of Natural Products, that we are hoping to bring it out soon. But this is where you see now Muraya conigi leaf. Once we see the phytochemistry, we have done LCMS, and to our surprise we see 16 peaks. Literature shows 22 carbazole alkaloids. We could see 16 out of that in our fraction, and that was identified by LCMS. So I am not showing other compounds. They are related structures. You know, always natural products, where is the complexity? Where is the problem? That their complexity. They are a complex mixture of closely related structures. This code itself you can see, except the quinine group in the first structure, that is not there. But then what we have a methoxyl group 1, 2 methoxyl group making different compounds, different polarities. Even there are compounds with same molecular weight but different structures, positional isomers, structural isomers. So all these things are actually part and parcel of the natural product extract, the complexity, and that's why it is not easy to develop a drug from this. But we have now means and technologies, tools to do it, and that is what you can see here. These 4 alkaloids we have isolated among the 11 in my lab. Different students have done the effort. So 11 to 12 alkaloids were isolated, and these are 4 we are using as markers, as well as the markers 3. Here they are responsible for the activity of alkaloid enriched fraction. That is also the part of regulation. That the activity of AEF you have to correlate with the markers, and we have correlated that with mahanimbin, coinimbin, and codolicin. These are the 3 compounds that are present. I have given the content of this percent bed by bed in dried leaf of all these alkaloids that you can see there. And this is where the effort has to be there. Now you see, this is the chromatogram. Now it is methanol extract chromatogram. So you will be seeing that, must be wondering when I am telling there are almost 150 compounds, but here you don't see. But you see there are so many small peaks in the baseline. They are not seen here. If you zoom in, you will find a very complex product. And this is the actually problem here. Unless and until you have HPLCMS, and good knowledge about the interpretation of the mass data that you generate and fragmentation, then you can identify the compounds online without isolating. Even if you don't have any standard, we can do it. So we never had any standard here. When we started work, we isolated the compounds and then used them as standards as well as done LCMS to identify the minor compounds where you may get new compounds. And this is another actually objective in natural product research, that to isolate new molecules along with the knowns. And this is another academic interest, I would say, but still that gives you a lot of credit and very high impact factor publications. That is where these minor compounds one has to look into. And these compounds then here that we have quantified also by HPLC, and that percentage I have already mentioned. And you see that here these are the 4 lab batches when we started before we go to pilot. That I think if I am able to show. So you see that here.
चलता है
Okay, so till the mic is getting. Okay, so you can see here there are 4 tracks. These 4 basically. 1, 2, 3, 4. These are the 4 tracks where you don't see the red color spots. Red color spots are actually these are, I can speak about Michael audible enough. These are the chlorophylls. Any leaf drug you take contains lot of chlorophyll. So you will see those 4 tracks are the alkaloid enriched fraction, where we have removed all the interfering compounds, and I say that there are 16 to 17 peaks major that we see in this fraction, which is being developed as pharmaceutical also, you can develop. So this particular process actually, it requires the different chromatographies using different resins. We have to do trial and error, and we use one of the resins called as HP20 diene resin, where we have actually made this alkaloid enriched fraction free of the polar compounds as well as chlorophylls. So this is how one can actually prepare the enriched fraction, and then you can formulate it into any therapeutically important or acceptable dosage form. Like with herbals generally we go for oral solid dosage form, so either tablet or capsule can be prepared. So we are already going for a capsule formulation. So we have actually identified the outsource capsule filling that we don't have in Nyper right now. From the company they will fill 50,000 capsules which will be required for clinical study, and then I think that is the way already we have validated the pilot scale trial for this formulation which will work in industry once they take up this. And also Curryleaf is a very important and easily available plant. So there won't be any issue of the availability of raw material, because we have to ensure that also. So that can be done through contract farming. So material is there, plenty of it, and the product can be developed very easily. So this is one example that how you can develop a drug or a product from a single medicinal plant, which is very well known, commonly available around us. So this is the, methodology. I will not go into detail that we are using in our lab, the cell-based assay. PT3L1 adipocytes, that we do the differentiation, free adipocytes to adipocytes, and see the cell viability before we go for real actually testing, because we have to ensure that the activity is not due to toxicity. So we have to do this MTT assay that we do in PT3L1 cell lines, and also the activity where we use oil red stain to actually do the staining for the, let us say, fat or lipid that is present in adipocytes. So all that lipid will take a red stain, and when the compound is having anti-adipogenic activity, the lipid level will go down. That way you will see there is a actually less dye coloration. So the coloration or dye coloration will become faint from dark red to light red if there is an anti-adipogenic activity. That is the observation you do, and you see here the alkaloid enriched fraction is more active than the individual 4 compounds. But that is what the important thing, that definitely there is a synergism here. They show better activity in combination after that, once they separated. So this is another indication why we go for the fraction, why not for the single molecule as the product development. And this is the, you can say, reason that you see here very well. This is the lipid accumulation study in this 3T3L1. And then this is the animal model that we use. All pharmacologists, some of them may be aware of this. We use high-fat diet fed in a mouse model of obesity, where we use this gray color small mouse, which weighs maybe, let's say, 80 to 20 grams. And then once you give them the high-fat diet, they will become bulgy. They will gain weight up to 38 grams. And then we have to see how much this weight reduction is there in once the treatment is given. And these are the, you can say, study line, this protocol. We have published this part in Journal of Ethnopharmacology already and filed patent earlier already that I have shown this way. So you can see at 100 mg per kg per oral, this was the most effective dose, where we have done the acute toxicity study, 28 days repeated dose acute toxicity study of this alkaloid enriched fraction, and no lethal effects were found during this 28-day observation, and no toxicity signs were observed in animals up to dose of 1 gram per kg per hour. Therefore, we can say this AF is safe at this tested dose. We can go ahead with that. And this is the biochemical parameters that you see: serum triglycerides, total cholesterol, serum low density lipoprotein, and high density lipoprotein. So, to our observation, to our surprise, that this particular fraction, it increases HDL. There is no medicine in the market anti-obesity that increases HDL. And this is another, perhaps, the attraction companies told me that that's why we want to go with this product, because it increases HDL. That was a very catchy observation. Industry took it, and that way, this is the first example. In my lab, we have 3 more herbal drugs. Even they are better than this, showing increasing the HDL. Now that is where I think we have the edge, and why the industry is attracted to develop such kind of products. So this is the very important thing we observed in our experimentation. And you see, this TG was lowered, TC was lowered, LDL was lowered, HDL is increased at this dosage that you see there. I have mentioned. We have compared also with per se dose and group also we have kept, apart from the disease control group that you see there. So this was the mechanistic study we have done with certain gene expression studies of fatty acid synthase, then PPR gamma, then we have used SRPP, 1C protein, and even adiponectin and ghrelin. Those were also studied. These are the important, you can say, genes that are upregulated in obesity. And you see that this particular area when given, they actually downregulated all these different genes that we have done the studies. And this work we have published out of the patent in Journal of Ethnopharmacology recently. So this is where what I want to show. You can develop the product from a single medicinal plant. Now, as I am speaking, we are preparing for clinical trial, and that is what in my lab. Now, a lot of work and activities going on that we are getting the formulation ready to be fed to the patients so that we get the ultimate result that we want. And let me tell you, we started this work in COVID time. No student was there in the lab, and let me say 2 technicians, and I was going in the lab even though there was lockdown. Being NIPER, we are actually a dedicated campus away from city and all that. We could go to the lab as well. I went every day to lab to COVID time. And then we started this work, and now 2025 we are almost here. So it takes a lot of hard work. Many obstacles came in the way, which I did not, of course, get time to tell, but you will face that. Unless and until you have that, you will not get the positive result without negative results. So people don't give importance to negative results. I think it is important also to learn from negative results and proceed on. So we had a lot of negative results initially. We repeated experiments, and all this data you see, it is not one-time data. Like I don't want to undermine the pharmacologist, Dr. Sharma is also pharmacologist, but one-time experiment is very dangerous unless and until you reproduce the data. Otherwise, it's only a publication you get and forget. That should not be happen. Whatever result I get, another oncologist should able to replace it. If we have done that, and we find there is a consistent observation with respect to HDL, I was very worried because I thought we have got whether others will got it or not, because industry will give your data. Let me tell you, industry, they want to see all the raw data also. Once they sit on the table, because they are going to invest money. They are not ready to invest money, and you know India generics. So generics companies are never interested in R&D, let me tell you. We have vast experience of interacting any company you name in India, they will never invest unless and until they really see that there is a potential, and they think academicians उनको दो पैसे ज़्यादा दे दो वो कुछ भी डेटा दे देंगे. That should not be the case. We said no. They said we will you come to our company, we will arrange everything, all the things, show your data. I said no. This is already under patent. If we should sign for CDA, confidentiality agreement, then only share data, otherwise no. That is very important need of an hour, that we should also show our importance to industry as academicians, researchers, and we do understand the economics. Not only they understand economics of developing the product. Unless and until you have this attitude, they will not listen to you, nothing will happen. We have a lot of this experience. With that now we have reached with this, because we tried several products earlier also that did not go through. Okay, so it's always like that. So this is the, you can say, product development from AEF, and these are my PhD students only, which are currently there in my group, and one scientist. That we have our natural product free laboratory in Naiper. We are very lucky that we have got 10 acres of land just for medicinal plant garden, and another 5 acres for cultivation of medicinal plants on the 140-acre campus that we have in Mohali, very close to Mohali Cricket Stadium, not the Mullapur one, the new one where now we see the matches, near stadium. We are very near to that, and this is our research group and field laboratory. In the entrance of that field laboratory, students have gathered there on one Saturday, and then they actually took the picture there, and this is where once you enter Naiper, this is just on the both the sides. We are entering from this side, we have the natural product research and medicinal plant area on both the sides of the Naiper campus, and then inside you go, we have auditorium, research blocks and all that. At Naiper Mohali, one of the greenest campus in Tri-City of Chandigarh, Panchkula and Mohali, I would say. This is Naiper Mohali. So next year, I missed the slide actually. Now, next year we are doing an international conference at Naiper Mohali under the banner of Society for Ethnopharmacology. And this is going to be an international conference where we expect 500 participants and 20 speakers from abroad and 30-35 from India. We will have a very fruitful and very intensive scientific conference on this, all this aspect of phytopharmaceutical, nutraceutical. We have an iConnect meet that is very important in the sector of, you can say, herbal drugs and natural products. So I invite many of you to attend this conference. Now I don't know, I will miss the slide. I have prepared a slide for announcement of this conference. We already started planning of that conference, which will be February 26 to 28, 2026. These are the dates. So if you are interested, anyway, I will share with Dr. Sharma the details he can pass on to the participants. So with this, I thank you all. If there are any queries, I will be definitely happy to answer this. Thank you.
Very interesting work, like the team is working in obesity also.
Yeah, obesity.
Anti-obesity.
Yes.
So, highlight some of the features of the gut microbiota in obesity and in non-obese. Because our lab is working in this area, like.
You are right. Yes, yes.
because gut microbiota plays a very important role. So, is there any difference in between the obese and non-obese? Like, if you have an
Yeah, we haven't yet looked at that, but yes, I think my lab is also working on gut microbiota, influence or the effect of these herbal medicines on gut microbiota. Let me tell you, even they act as prebiotic. We have data on Trifala. Trifala, so normally in one of the PhD students is working with in collaboration with Nabi Mohan. They have the fermenter and all this model. So they have shown that many of our these herbal medicines can act actually as prebiotic. So definitely once we talk about in obese and non-obese, we should see that we haven't seen that angle, but yes, it is worth checking. Definitely this gut microbiome has an important role to play even in obesity. So we need to check that. We haven't touched that part. But yes, we have done work with Trifala. I can tell you it is definitely converting. You see, Trifala has cannins. All these hydrolyzable cannins, they are converted into urolithins. And urolithins are the metabolites that are responsible for the good health, promoting good health of the bacteria, gut bacteria. And that we have shown with bifidobacteria as one of the major colonies of this gut microbiome. So that work is also going, but this was not the point of this session. But yes, definitely, it's worth to look in that aspect. So, any other faculty member, any question? So somebody working here in pharmacology, in faculty? No. Here, among the participants?
HOD of pharmacology department. She is an examination.
Okay.
Diploma
No,
exam.
maybe in the participants I thought it's somewhere. But see, why I'm asking this, this is very important discipline. And I mean, people should try to work in collaboration. Even you are in pharmaceutics, I do a lot of collaboration in pharmaceutics in Pharma. So many of the formulation aspect, no, because we are not doing directly, but yes, our colleagues they are there to help, so we can do like I am using regularly Zeta Sizer. Okay, that Sutix people need, then different instruments with respect to the further particle size, 10, all those equipments we have. So it is better to have this collaboration because now there is a dearth of new APIs. We don't have any drug molecules right now. So what people are doing in research, they are going towards all phytomolecules. Many pharmacologists are working on, let me tell you, let us say pinostorm. This is a flavonoid present in, turda. That plant leaf, it contains actually pinostorm and several flavonoids. I mean, people are working on that, lignans as well as stilbenes. So these are the molecules which are, even epigallocatechin gallate present in green tea, major polyphenol, they are working on them. They don't have molecules. So it's a good time actually. We should have a very nice, like we are working on manning, for example, like several others in the lab, like pyradine we work on, and so many different, we have a number of molecules from plants. If you just select shortlist 10 molecules, you can do 10 years research on it, one molecule per day. So that kind of opportunity I think we have for the faculty, those who are thinking what we can do in pharmacognosy or natural products, we can do a lot. That is what, so every science in pharma, I think, it starts with natural products. Medicinal chemistry actually emerged from natural products, none than any other field. When we see, I studied in Austria, Germany, so there we see very strong organic chemistry because they actually are the people who actually develop this field of organic synthesis. And then I think we see India also has a lot of potential. And India's, in fact, potential is known in organic chemistry, but still, you see, we rely on China for our APIs. And this is the now hot topic these days, that we have the KSM, key starting materials for these APIs that we work in NIPER as well. So these are the areas, then why can't we have APIs from herbs? We have a lot of potential. So this is another new dimension that we are working with current ministry, and even we form now, we are forming Section 8 company. This is very important, like institutions who are interested in developing their product, you must have Section 8 company in your institute so that industry can come and collaborate, talk with you. So you can find out the details. So these are the things I think which we need when we talk about educator. We have to educate these students who want to become an entrepreneur. So for the students, there are very important things that we should tell. Apart from the textbook curriculum, we must tell all other these related things to our students. And that is, I think, I suppose the big responsibility as a teacher we have. And that is what we try to do at NIPER. This love curriculum is all research based than simply pure theoretical based. So that is what I think all the institutions should do. IITs, why they are successful. Their curriculums are based on research, based on the practical applications, commercializations, technology transfers. We don't do in pharma, but now we are doing. I mean to say the education institute, education part. There are R&Ds in India, but not the basic drug discovery research is done. Even today, it's not done. So these are the issues, but that is what I want to tell. Few points which I thought apart from. If you don't have queries, so I thought you think on these lines and try to see what can be. Okay, so with this, thank you. I took exactly 1 hour, 11 to 12, almost. Thank you.
Thank you so much, sir, for your insightful talk on nutraceutical and phytochemistry. Your words painted a canvas of wisdom. Thank you so much for sharing this valuable experience and enlightening our mind.
Now, I would like to request Mrs. Neha Gupta to come on the stage and give a brief introduction to our second speaker, Professor Pallavi Ahirao, APTI National Co-Convener, Women Pharmacy Mohali. Good afternoon to one and all present here. I, Neha Gupta, Associate Professor, Pharmacology. It is my honor and pleasure to introduce our esteemed guest speaker for today's FDP, Dr. Pallavi Ahirao ma'am. Dr. Pallavi Ahirao, working as a Professor of Pharmaceutical Chemistry in Chandigarh College of Pharmacy, Landra, since last 12 years. She has total experience of 17 plus years in teaching and research. Her field of specialization include isolation and characterization of phytoconstituents from natural product, formulation and development of nanoparticle, NLCS, antimicrobial resistance, and evaluation of efflux pump inhibitory activity from natural product. Toxicity study, various pharmacological activity such as anti-inflammatory, immunomodulatory, cytotoxicity, anti-rheumatoid, anti-obesity, etc. She has to her credit 25 international publications include 2 book chapters in CABI, UK and Springer Nature Publisher and 3 granted patents. She also has authored 1 book of organic chemistry. She has attended more than 75 conference, seminar and workshop and 3 certificate course from JCR, London School of Hygiene and Tropical Medicine from University of Cambridge. She has been called several times as a resource person in FDP, workshop and conference. She is a life member of various associations including Indian Pharmaceutical Association, Association of Pharmacy Teacher of India, Society of Pharmaceutical Education and Research, Indian Hospital Association. Dr. Pallavi is a national co-convener of Women Forum, Association of Pharmaceutical Teachers of India. Apart from her academic career, she is actively involved in Women Forum, Association of Pharmacy Teacher of India, APTI, and conducted several scientific webinars, workshop and seminar with audience of Pan India. May I now request ma'am to kindly come forward and grace the stage.
Good morning now, all of you. So after we invite Professor Sanjay Jachat's lecture, which was very thorough and enlightened us with vast knowledge on natural products. My area is also on natural products, and I work on efflux pump inhibitors out of many several areas like anti-obesity, anti-hematite, and anti-inflammatory, cytotoxicity, as well as toxicity studies. So before going on my topic, because my introduction was given in like I National Co-Convener of Women Forum of APTI, that is Association of Pharmacy Teachers of India. So I just would like to know how many of you are members of APTI? 1, 2, 3, 4, 5, 6. Okay. So I would request Professor Saurabh Sharma sir, you can go for group membership program, sir, wherein there is a concession given because life membership fees is 5,000, but 3,500 is the for that concession is given if more than 5 members are there. Okay, so because this APTI is only the forum wherein for pharmacy teachers, because we don't have any other kind of forum wherein we can put our grievances in. So that is why, and it is also required because we are in academics that at least one body should be there where we should be a part of it. So I request all of you to be a part of APTI, and under this APTI Women Forum, especially for women, we conduct so many webinars, workshops, summer courses, faculty development programs, and even conferences in various colleges pan-India. Okay, so I have seen that there are 5 zones like north, east, west, south, and central zone. But in north zone there are total 7 states are there, including Delhi NCR, Uttar Pradesh, Himachal Pradesh, and Punjab, Haryana. But in Himachal Pradesh, there is no coordinator or convener of women forum. That's what we noticed. In rest other, all the states, all the like in women forum, the central committee as well as state committee, they have the convener as well as co-conveners. Okay, so now I start with my topic, antimicrobial resistance, and before that I would like to thank Professor Saurabh Sharma sir for giving me this opportunity and inviting me for this faculty development program on empowering educators for excellence in academia and research. And the area that is antimicrobial resistance and natural products as a potential source of bacterial influx from inhibitors. So natural products, the work already has been given by Professor Sanjay Jachak, that how vast is the area and why natural products only, because we gave so many phytoconstituents, so many chemistry, the diverse chemistry, which is just not possible if we are synthesizing it in the lab. So I will tell you in a very easy manner that how you can go or proceed for your research work in natural products. Okay, so for that what you need to do. So one by one, as I proceed with the lecture, I will tell you how I proceeded with this part. Okay, so the topic is antimicrobial resistance, and as antimicrobial, we know that the microbes, that is the bacteria mainly, are responsible for life-threatening diseases. And these microbes are very smart entity, and whenever a drug is given, antibiotic is given, that antibiotic they just flux it out. That we call it as efflux pump. Now here in this slide you can see that these are the bacteria, and on the cell wall there are embedded protein pumps, and these protein pumps we call it as efflux pump. Efflux means efflux out. So whenever antibiotic is given for the treatment of bacterial infection, that antibiotic may not retain insufficient concentration inside the bacteria to kill it, because as I said, bacteria is a smart entity. It will immediately take out the maximum concentration of antibiotic out of the cell wall, out of the cell, and hence we call it as efflux pump. So what if that efflux pump, if we block it? So if the efflux pumps are blocked, then sufficient concentration will remain inside the cell and bacteria will get lysed. Okay, so that is the work I have worked on this part. So this is a very simple diagram which shows how efflux pump inhibitors will act. Okay, so here you can see the red part on the arrow. That red part, that is a drug actually or a compound which will block the efflux pump in such a manner that the efflux will not take place. Okay, so now why antimicrobial resistance? Why I have chosen antimicrobial resistance as a part of research? So one of the reasons is that it is growing exponentially and it is the most urgent health crisis. So it is said that like by 2050, death of 1% every 3% will occur. And what are the factors for this antimicrobial resistance? So numerous factors are there, like population explosion, industrial expansion, ease of global migration, or over-the-counter access of antibiotics. That is the most prevalent one, I would say. Rapid emergence and spread of resistance pathogens. Apart from that, imprudent usage of antimicrobials, which aggravate the threat of AMR. And then another one is the enrichment of AMR genes. Now this is very, very important, and it is the present in bacterial population due to rampant antibiotic usage in hospitals, agriculture, poultry and animal husbandry. So from 2000 to 2010, the usage of antimicrobials increased by more than 30% in low, medium and income countries. And the major contributors and due to prevalence of infectious diseases and also because of the easy availability of antimicrobials. And these are available or spread in both community as well as hospitals. And for the treatment of microbial resistance, antibiotics are given. And what antibiotics are? So antibiotics are nothing but these are chemical entities which are responsible to kill the bacteria in various ways. Either they will act on cell wall or on genes or on ribosomes, or it will affect the protein synthesis, etc. And the main bacteria which are responsible to cause antimicrobial resistance are called escape pathogens. We call this escape pathogens. It is a group of 6 pathogens. Out of these 6 pathogens, 4 are gram negative and 4 are gram positive. So the escape E stands for Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. Now you must know that escape pathogens, because many of the time people they do not know what escape means. So escape pathogens are responsible majorly for antimicrobial resistance. And if you see the structure of bacterial cell wall, so gram-negative bacterial cell wall is more complicated and complex than gram-positive one. Then, so efflux pumps, you are aware of efflux pump now? These are situated and imbibed on the cell wall of bacteria, and उनका काम क्या होता है? एंटीबायोटिक को efflux out करना. So these efflux pumps are the lethal weapon bacteria possess to develop resistance. And because of this, one of the mechanisms for resistance is presence of EPs. And multi-drug resistant escape pathogens. So as I said, like Staphylococcus aureus is considered as the multi-drug resistant bacteria. Okay, whatever antibiotics are there, whatever class, maybe aminoglycoside, tetracycline, penicillin, beta-lactams, etc. So these Staphylococcus aureus will develop resistance. Especially the first resistance was developed, was reported in 1990 to tetracycline. So now what is the mechanism, antibiotic resistance, how this happens? So like here you can see, one of the mechanism is efflux pump, but apart from that, the second mechanism is maybe like inactivating enzymes are present inside the cell, which will inactivate the antibiotic. So whenever antibiotic is given, some chemical modification may take place and it will inactivate that antibiotic. Second is like it will alter the target site. If the target site is a genome, or if a target site is ribosomes, then it will alter that target site so that antibiotic, though it reaches it, it will not show its action. So that is the mechanism. So antimicrobial resistance can be of two types, molecular class and acquired means the emergence of resistance in bacteria by acquiring a drug-resistant gene. And it may be because of mutation or because of transferable drug resistance. And the acquisition may be temporary or permanent. This acquisition one of the reason. Second reason is inactivation or modification. Third is genetic elements, that is such as plasmid, ribosome. Next is alteration in the cell wall composition, so it may happen. And third is, the next one is the porins, cell wall porins. The number of cell wall porins which are present, it may decrease so that antibiotic may not enter inside the cell. So that could be another reason. And the last one is overexpression. So all these few mechanisms are given here of antimicrobial resistance, and out of which we will focus only on the efflux pumps. So in early 1990s, efflux pumps were reported in bacteria and found to be the new targeting microorganism. The tetracycline resistance was first reported in E. coli, and classification of efflux pump. Now this is again very important. Classification: gram-positive bacteria, gram-negative bacteria, escape pathogens. You all are aware now. These efflux pumps are classified into 5 superfamilies. The first superfamily is small multidrug resistance family. Second is ATP binding cassette, that is ABC superfamily. Next is MFS, that is major facilitator superfamily. MADE family, that is multi-drug and toxic compound exclusion family, and resistant modulation division family, which is found only in gram-negative. Rest of the 4 families can be found in gram-positive, gram-negative, but RND is found only in gram-negative. So these families, क्या है ये families? So I will show you here in this diagram now. Now, this is, all these families are there: MADE, MFS, SMR, RND, ABC. These are super families, and these are efflux pump. कौन से हैं? MADE में है MET A, MFS is NOR A, SMR set A, RND MEX B, and ABC is LMR-CT. So these are efflux pump. And what efflux pump actually are? These are nothing but these are proteins. Proteins are made up of amino acids. So protein structure which is imbibed here are responsible to take out certain drugs. Say for example, if I take an example of MFS family, which is a very prevalent family and present in Staphylococcus aureus, and NOR-A efflux pump is overexpressed in it. Overexpressed means majorly it is present. So in Staphylococcus aureus, MFS family, NOR-A pump is overexpressed. And what NOR-A pump efflux out? Mainly fluoroquinolone, norfloxacin. Can you see this? FQ, NF, EC. All these are the drugs or the compounds which these pumps are effluxing out. So fluoroquinolone, norfloxacin, tetracycline, oxazoline, chloramphenicol, berberine, and ethidium bromide. So ethidium bromide is a dye, and that is, this NOR-A pump take out these drugs out of the cell. This diagram is clear? Okay. Next is methicillin resistant Staphylococcus aureus is responsible for both community and hospital acquired infection. So there is a Staphylococcus aureus and there is methicillin resistant Staphylococcus aureus. Methicillin is an antibiotic, maybe beta-lactam antibiotic, and that methicillin is resistant, is shown by Staphylococcus aureus, and we call that strain as MRSA, that is methicillin resistant Staphylococcus aureus. And in that, NOR-A is overexpressed in MRSA, which is responsible to take out the drugs like ethidium bromide, rhodamine, acridine, and various quaternary ammonium compound, etc. So here you can see that reserpine, verapamil, ethidium bromide, rhodamine, and acridines. These are the compounds we take actually as a standard or to compare our drug test compound. Okay? So we consider it as a target compound. So you know verapamil, you know reserpine. So these drugs could be act as a standard or standard target. Okay? So why this drug, that I will show you in next slide. So NOR-A pump is composed of 388 amino acids and with 12 transmembrane segments, and it belongs to MFS family, and MSRA belongs to ABC family. So why I am telling you only about NOR-A, because on NOR-A pump only I have worked on. Now efflux pump and its clinical relevance. So both bacteria and eukaryotes possess efflux pumps, and it can confer MDR resistance. They can add themselves to other resistance mechanisms this way. So this is intracellular infection, and apart from efflux pump, these are also responsible for other resistance mechanism also. So synergistic mechanisms it may follow for resistance. And hence EPs can be seen as a new target. Moving on to efflux pump inhibitors. So an efflux pump inhibitor will restore the activity of the drug resistant cells. One, because once it is blocked, antibiotic concentration will remain inside the cell and resistance automatically will go down. Then it may be devoid of effect with wild type cells. It decreases the frequency of resistant mutants. So these are the efflux pump inhibitors parameters we can say. And EPI would act by binding to the pump with increased affinity, impairing access to the binding site, and dissipating the energy source. So these are the mechanisms by which EPI would act. Then why EPI as antibiotic potentiator? So EPIs are the compounds which will block the efflux pump, and these are need to be given along with the antibiotic. There are certain things which when we are going for EPI, so first is that it is one of the most promising approach to tackle the crisis of antibiotic resistance. Reversal or removal of acquired as well as intrinsic antibiotic resistance, and it can be achieved via inhibition of drug transporters. It reduces the frequency of resistant mutants